Good morning, and welcome back to the general scientific session. We are going to proceed with our first speaker, Dr. Karim Zaghloul from the NIH, who will be speaking on advancing the neuroscience of human memory through neurosurgery. He's been doing amazing work using invasive physiology. Looking forward to it.
Okay, thank you for inviting me to speak here today, and thank you for giving us the opportunity to present our work, and of course thank Russ and the organizers for putting together a great meeting. What I was going to focus on today is how we can use neurosurgery as an opportunity to advance the fundamental neuroscience of memory. And before I begin, I want to state that I have no financial disclosures. So everyone here knows, of course, that memory is critical to our everyday experiences. We rely upon our memories not only to form our own personal identities, but also to imagine and to guide future actions. And memory, of course, is also highly relevant for our specialty. When we perform our surgical resections, oftentimes one of the hardest questions that we have to answer is, how will this surgery affect my memory? And for that, we don't have very clear answers. Now, the study of memory has spanned thousands of years, but broadly speaking, you can think of the human memory as being divided into several subsystems. So there are things like procedural memories, the memory for how to tie a knot, which is part of the implicit memory system, and semantic memories, memories of facts and concepts, which are part of the declarative memory system. But what I'm going to focus on today is this special type of memory known as episodic memory. This is the memory of episodes or events that occurred at a specific time or place in the past. So for instance, it's a memory of your first date, your last vacation, or a specific patient interaction. Now, it's argued that humans are, the study of episodic memory is best studied in humans. Humans are unique in their ability to use language, and so they can explicitly describe a recollection, which is one of the requirements of episodic memory. Now, one of the key features about episodic memory is that when you recall a memory, you actually internally, in your own mind, travel back in time to re-experience it. And the idea goes like this. What I'm showing you here is a very, very simplified version of your brain, and your brain exhibits these fluctuations in the state of the brain. And these fluctuations are influenced by who you're talking to, what you're seeing, and so on and so forth. Now, suppose that you experience an episode, suppose that you and I have a great conversation at a restaurant. If you're going to remember that conversation, you're going to embed the memory of that conversation within the context of this brain state. Now, suppose at a later point in time, you're walking down the street and you see this restaurant. Seeing this restaurant will trigger you to remember that conversation. Well, there's substantial behavioral and psychological evidence that suggests that what happens is that you actually, in your own mind, internally jump back in time and re-experience not only the episode, the conversation, but also the context of that conversation, the restaurant, the time of day, and so on and so forth. And everyone here knows this is true. You know, for instance, that when you remember or you think about the first time that you met your significant other, you can go back in your own mind and exactly play out what happened, what you were wearing, what time of day it was, and so on and so forth. And so the question is, how does this actually happen in the brain? How does the brain actually allow you to perform this process of mental time travel? And what I'm going to show you today is that by using the signals that we can record from the brain, we can actually find evidence for how this works. And our hypothesis is that if we look at the activity that's happening in the brain at this point, when you actually experience an episode, it's possible that that same activity is actually reinstated or replayed when you remember it. So in the study of memory, we have a lot of tools that we use. We have things like functional MRI, and we have lesion studies, and this gives us a good sense of where things are happening in the brain and where things, or what things might be critical for certain processes. At the other end of the spectrum, we also can use animal studies, and we can look at individual neurons. So we can see, for instance, how neurons might change their synapses during learning and memory. And then there are a number of studies looking at the activity of neurons in the hippocampus, in rats and in mice. And you can see this activity changing when a mouse navigates, for instance, a spatial environment. Now, importantly, that activity is actually replayed when the mouse either consolidates that memory or thinks about that memory to try to make a decision. And so what we have here is we have information that occurs, or that we have at the very largest spatial scale and at the very smallest spatial scale, but we have this gap in between. And the question is, how can we fill in that gap? And this is where I think neurosurgery can play an important role. We can use our recordings to actually understand how we can move from the activity of individual neurons to circuits and local populations to larger networks. And the reason we can do this is that we record activity directly from the brain, primarily in the patients that we treat with medically refractory epilepsy. So as you know, in order for us to offer these patients the best chance of seizure freedom, we have to accurately localize exactly where seizure activity is coming from. And to do that, oftentimes, we'll place electrodes either directly on the surface of the brain, like these electrodes that you see here, or deep in the brain using depth electrodes or stereo EEG electrodes. And one of the advantages of this paradigm is that while they are actually being monitored, these patients are awake and they're interactive, and we can ask them to participate in different types of tasks that are designed to probe their memory. And we can see what behavior looks like compared to the physiology that we record. So if we look at these signals from any one electrode, we can actually capture the activity of what's happening in a small region of the brain. We can also look at coordinated signals across multiple electrodes, and we can get a sense of what's happening at a larger network level. And then more recently, we've actually been able to develop an approach for capturing or using microwires to capture the activity of individual neurons. And so it's these three spatial scales, looking at this activity in the human brain at the neuronal scale, at the local scale, and at the network scale, which allows us to begin answering these questions. And what we want to know is activity at these three scales providing us evidence for how it is that we perform this operation of mental time travel? So I'm going to start with the middle scale, this local scale. This is a scale, of course, where neurosurgeons have already been playing a leading role. We look at local activity, for instance, to understand how humans process language and speech, or how we can visually discriminate different stimuli. This is the work of many groups around the country and around the world, including here, Eddie Chang and Jeff Ogeman and Matt Howard and many, many others. And what we're going to do is we're going to look at how this local activity looks like in the context of memory encoding and retrieval. So we have our patients play these little games. This is a game that we call the paired associates task. So in this task, they actually are given two pairs of words. They're supposed to remember that these words go together, and then at a later time, we cue them with one word, and they're supposed to recall the associated word. And forming associations and then using those associations to actually drive your memory is something that we do routinely. And so that's one of the reasons why we chose this task. Now, one of the advantages of this task is it actually gives us direct control. It gives us control over what they're remembering or what they're encoding, and it gives us control over what they're trying to retrieve. And so we can see what the activity looks like at both of those periods. So let's first start by looking at what's happening during encoding. We can look at the activity at all these electrodes every time that they're encoding these pairs, and we can ask, what does the activity look like when they're encoding things correctly versus when they're not, and are there any differences? And if we do this in any one patient and we aggregate it across several patients, we can create these very high precision spatial temporal maps of what local oscillatory activity looks like when you are encoding something successfully in your brain. And what you should see is that we have activity that starts generally in your posterior temporal lobe and then migrates over to your left dorsolateral prefrontal cortex. And so this is what happens when you're encoding something. It turns out that something very similar happens when you're retrieving, and we can actually quantify this. We can look at the distributed patterns of activity across all of these locations during encoding, and we can ask, are they similar to the patterns that we're seeing during retrieval? And when we do that across all of our patients, we can generate what we call these reinstatement maps. I'm going to walk you through it because this map is actually providing us the evidence that we're looking for. What we're looking at here on the y-axis is every single time point during encoding, beginning with when the words come on the screen. On the x-axis is every single time point during retrieval until they vocalize their response. The color tells us how similar that pattern of activity was between any encoding and any retrieval. And what this is saying is that when they are correct, immediately before they vocalize their response, there is a pattern of local activity in the brain that is quite similar to what the pattern looked like when they were encoding. We don't see this during the incorrect trials. And so this provides us our first piece of evidence, that if we look at the level of local oscillations in the brain, what we see are patterns of activity that are exhibiting reinstatement, and it helps mediate this process of jumping back in time to retrieve a memory. So now that we know this is true at the local level, let's look at the different spatial scales. So let's go now to the network level. And of course, understanding networks and connections has been really an exciting and significant area of interest for the last decade, and there are several neurosurgeons, of course, who are involved in these efforts. Now, oftentimes we use imaging to identify connections. We use things like DTI imaging, which can identify structural pathways in the brain. We use functional MRI imaging to identify correlations. But just because we find a structural pathway, it's not entirely clear whether that pathway is actually being used for communication. And just because we see correlations, it's also not clear whether those correlations represent true communication between brain regions or whether it's just a statistical relationship. And so in our lab over the last couple of years, we've actually been developing a technique to actually use the physiology to identify what are known as effective connections. Effective connections are a connection between two brain regions through which information is conveyed from one point to another with a specific time delay. It tells us how information actually moves in the brain. And so what we can do is we can take any person's electrodes and we can create a network map. So each of these nodes represents the electrode location in someone's brain. The connections between these nodes represents the presence of an effective connection. And as I said, the effective connections tell us how information moves. They each have a direction of movement designated by this arrow. And what we can see is that if you look at the average direction of movement in the temporal lobe in one participant or in all our participants, what we see is an average flow of information that's moving along the temporal lobe from the back to the front. And this is consistent with what we saw in those earlier movies where we see this propagation of activity moving in this direction along the temporal lobe. And so now we can ask what's happening in these connections and communication between these connections during encoding and retrieval. What I'm showing you here is a pair of electrodes, and you have to understand that when two brain regions are connected, they're not actually always communicating. Sometimes they're communicating and sometimes they're not, and this makes sense. This communication is actually dynamic. We can actually capture the dynamic nature of this communication, And we can see what it looks like when they encode things successfully versus when they do not. And what we see is greater communication during the correct trials in this particular pair compared to the incorrect trials. And we see a similar pattern when they're retrieving correct trials. And again, we can actually quantify the extent to which communication now is being reinstated among all these different connections. And again, we create these reinstatement maps. And again, we see that there is significantly greater reinstatement, this time in the amount of communication that's happening along these connections between encoding and retrieval. We are using this communication to jump back in time and to retrieve these memories. And now finally, I'm going to talk about the smallest spatial scale. This is the neuronal scale. This is, of course, an area of intense research and interest. And this has been primarily captured through these microwires that extend out of these depth electrodes. These depth electrodes are placed in the MTL, for instance, and there have been previous studies that have shown that neuronal activity in the hippocampus, for instance, shows specific responses to visual stimuli, or also participates in spatial navigation of a virtual reality environment. This is, of course, the work of Itzhak Fried, and Sharon has been involved in the work, and many, many others. More recently, we've been actually using these microelectrode arrays actually placed on the cortical surface. This is an approach that was pioneered by people like Guy McCann and Imad Iskander, and more recently Samir Sheff. And the idea is to place these arrays directly on the surface of the brain. And one of the advantages about these arrays is it allows us to capture the activity of a population of neurons. Each of these electrodes might actually capture single unit spiking activity from individual neurons. And in fact, that's exactly what we're looking at. So here's one trace from one electrode. We see single unit action potentials from one neuron, and we can see what these neurons are actually doing during memory encoding and memory retrieval. So to show you what these data look like, what I'm showing you here are two example neurons. And what you should notice is that there's heterogeneity in the response. There are some neurons that show decreases in their spiking activity, and there are other neurons that show increases. And actually, this is exactly what we'd expect. We don't expect all the neurons to be doing the same thing. In fact, if we look at all of the neurons from one array and we line them all up and we see what's happening during the encoding period after words come on the screen, which is indicated by this dashed line, we can see what the spiking activity looks like, which is indicated by this color. And so you can see that there are some neurons that are increasing their spiking activity during the encoding period, and there are other neurons that are decreasing their spiking activity during the encoding period, and we see this heterogeneity. Now, remarkably, we can see what these same exact neurons are doing when people are retrieving these memories. And what we find are very similar patterns of activity. The neurons that increase their firing during encoding are increasing their activity during retrieval. The ones that are decreasing their activity during encoding are decreasing their activity during retrieval. They're doing the same thing. And now we can quantify this again, and we can use our approach for generating these reinstatement maps. And what we find is if we look across several patients is that if we look across the population activity in these arrays, we see significant evidence of reinstatement. The neural activity itself is being reinstated, providing us our final piece of evidence that at the smallest spatial scale, we see that the brain is trying to replay these signals. So in conclusion, what we've done and what we've been able to do because of our neurosurgical recordings is actually investigate the brain across these spatial scales. We can see what's happening at a network level, we can see what's happening at a local level, and we can see what's happening at the neuronal level. And in each of these spatial scales, what we're finding is evidence that the brain is actually replaying or reinstating neural signals, which allows someone to essentially jump back in time and remember a particular episodic memory. So in conclusion, I want to thank, of course, all the current and former members of my lab who made all this possible, our very close collaborators on the epilepsy service at NIH, and my partners and collaborators in the surgical neurology branch. Thank you.